TriCep is established by URW and is supported by the Australian National Fabrication Facility through the Materials Node. TriCep is established to facilitate the translation of fundamental advances in 3D bioprinting into real clinical solutions. As such, we work alongside clinicians from around Australia, indeed around the world. These clinicians and collaborative projects include Professors Peter Chung and Claudia DeBella, working on cartilage regeneration in the knee, Dr. Payal Mukherjee on 3D printed ears, Professor Toby Coates on the creation of 3D printed scaffolds to improve the efficacy of islet cell transplantation to treat diabetes, Professor Gerard Sutton in pursuit of the bioengineered cornea, and Professor Fiona Wood developing 3D bioprinting protocols for skin regeneration, and in particular, the treatment of burns. Each of these endeavours requires the development of customised bioinks and bioprinters in a way that ensures seamless transition into a larger scale manufacturing environment when required. Each of my colleagues will now tell you how we are achieving that at TriCep.
I am Sanjeev Gambhir, Associate Director responsible for bioink development at TriCep. We have a team of skilled chemists and engineers engaged in developing and formulating a wide range of bioinks. These inks are comprised of materials that are compatible with living cells. They protect the cells during printing and ensure appropriate cellular development after printing. The ability to print 3D structures wherein bioink compositions and different cell types are strategically distributed to affect tissue regeneration has been clearly demonstrated. These multifunctional biomaterial compositions are critical to success in 3D bioprinting. As evident from increasing number of publications, rising from 99 in 2016 to 1700 by 2021, this is an area attracting great deal of attention. At TriCep, we want to focus that energy into the development of bioinks that are translatable. We work closely with primary industries, including the Australian seaweed farmers, to source the materials required for formulation of high-quality bioinks. At TriCep, we are developing customized bioinks and we have established the state-of-the-art facility required for scale-up. The quality control laboratory is equipped with all key analytical tools needed to ensure our end product comply with the highest quality standards. At TriCep, we have developed sterilization procedures for a range of bioinks and carried out shelf life studies to establish optimal storage conditions. We have developed packaging and delivery protocols to ensure that the high-quality product that leaves TriCep reaches your laboratory.
We have integrated a suite of advanced fabrication tools to enable us to develop biofabrication systems and workflow protocols to meet our customers' needs. As Associate Director of TriCep, I'm delighted to lead an experienced team of design, mechatronic, software engineers in providing innovative, practical solutions to biofabrication hardware, medical device challenges. The team are adept at working with SMEs as they embark on their commercial journey, working closely with all research, regulatory and commercial stakeholders to deliver products ready for deployment and use in a clinical environment. Developments include the Excelda BioPen for cartilage regeneration, the 3D ALIC hybrid printer for printing ears, and the 3D PICT to create structures for islet cell transplantation. Our experience with flagship biofabrication technologies and understanding the needs of clinicians has allowed us to identify gaps in the current market, where we now strive to assist in the development of the next generation of biofabricators through our research and education bioprinting platform, the 3D Ready. The 3D Ready, with a starter kit containing bioink formulations, has been supplied to partners across the globe, including Australia, Japan, Korea, Finland and Italy.
The TriCep team is committed to the implementation of quality management systems that facilitate compliance with ISO 13485 and GMP accreditation in order to provide the customised support needed to facilitate the translation of bioinks for 3D bioprinting from the lab bench to the clinic. As the manager of the ANF Materials Node, I work to ensure TriCep has access to the most recent advances in materials processing and fabrication. As Associate Director of TriCep Operations and Engagement, I facilitate supply agreements, operations, facility management, project contracting, engagement and reporting. As a first point of contact, I can direct your inquiries to the right person to ensure your project is completed in a timely manner. Our goal is to support your needs and deliver on your goals.
The TriCept team, supported by the University of Wollongong and AMF, has in place the expertise and facilities to help fast-track your ideas in 3D biofabrication. We can assist in the development of customised bioinks and 3D bioprinters. Our established clinical network and links to others with expertise in health economics and mapping the regulatory pathway provides insights that can set projects up to be translation ready. So please, do not hesitate to contact Pete, Sanjeev, Steve or I if you have any questions.